Punnet square notes. This is notes number 2. Let's get started here. So, first things first, let's talk about what Punnet squares are. So we've been talking all about these genetics and how about traits are inherited down, but one way that we really use to help us predict offspring genotypes is these Punnet squares. So if we know what the parents' genotypes are, the Lerda combinations, we can use those for specific traits to figure out what the offspring, the chances that the offspring will have those traits as well. We call these generally a monohybrid cross, or the ones where they only deal with one trait. We will talk about the other types in the future, but just know that if we're looking at one singular trait, called a monohybrid cross. And so from the genotypes, from the letter combinations, we can determine what the phenotypes of the offspring are going to be. So from the letters, let's say we had a big T, little t, that would be the genotype. We could determine that the phenotype would be tall. So our problem here is we're looking at Mendel. He is crossing a heterozygous tall plant with a short plant. And we know that tall is going to be our dominant trait. So first thing we need to do is write our symbols for each trait. So tall, in this case, is going to be dominant, so we are going to use the capital T, versus short is going to be recessive, and so we are going to use the lowercase t, as we can see right there. Step number 2 is we need to figure out the genotype of each parent. So again, we know that we are crossing and having a heterozygous tall plant. So heterozygous, remember, means one big letter and one little letter, or one dominant and one recessive trait. And then our short parent, short is going to be the recessive trait. And so the only way that we get that is if we are homozygous or have two small t's. So there's our two parents. Step number 3, we pick one of the parents and we write that along the top. So in this case, wrote the heterozygous individual up at the front. And then our second thing that we do is write the second parent along the side. From there, anything at the top comes down. So we can see this T is going there. We can see that T is going there. Little t goes there. Second little t goes there. And the other important thing to note here is that we always want to keep the capital letters first. After we bring the ones down from the top, we also want to bring the ones down from the side. So as we can see here, that T goes there as well as there, and that T goes there as well as there. And then we get our 4 possible offspring types. So let's look and answer these questions here. How many of our 4 results are heterozygous and dominant? Well, hopefully if you look over here, you can see that 2 out of the 4 are going to be heterozygous. How many are recessive? In this case, again, 2 out of 4 are going to be recessive. So 2 little t, little t. Now let's look at the genotype ratio. So genotype, that is going to be the letter combination, and we are going to determine what the ratio of that is. So if we look over, I see that we have big T, little t, and little t, and we have 2 of each of those. So our genotype ratio is going to be 2 heterozygous to 2 homozygous recessive. Our phenotype ratio is the ratio of how they look. And so that's essentially saying, what does big T, little t look like? And that's going to be 2 tall. And what does little t look like? And that's going to be 2 short. So again, we have 2 heterozygous tall possibilities to 2 homozygous short individuals is our phenotype ratio there. Let's do another example here. So we have a heterozygous tall pea plant crossed with a heterozygous tall pea plant. So first thing we got to do is write down our letters, figure out what is capital and what is lowercase. And in this case, we're using the same as before. Capital T is tall, lowercase t is short. Now we have to figure out what the genotypes of each parent are. Well, both of them are heterozygous, so we should have big T, little t for both. Then we have to go in and draw our Punnet square. So let me draw that for you here. Let's put one parent on top. And I'm just, for sake color-wise here, going to already bring those down. And let's do our other parent on the side. I'm going to bring those over. T and T. So let's look at our ratios here. So what are all the possible genotypes that we have? Well, we have big T, we have big T, and we have little t. And now we simply just have to count. We have 1 to 2 to 1 is our ratio of genotypes of letters. Then we have to look at our ratio of phenotypes. So what does big T tell us? That tells us that this offspring is going to be tall. What about big T? Same thing. We have at least that one dominant letter, that one capital letter. The only time we get short is if we have little t. So even though our genotype ratio is 1 to 2 to 1, our phenotype ratio is 3 tall, which are going to come from these 3. Get a different color here. These 3 genotypes here are all going to give us tall to the one short little t. Another practice problem here, because who doesn't love practice problems? So, different plant here, different idea, different trait. We are now looking at wrinkle seeds and round seeds. Round is going to be dominant over wrinkled. So, another key word here is pure. So let's first start out with our combinations. So round is going to be dominant, so we're going to use big R for round, little r for wrinkled. Now let's figure out the genotypes of each parent. So we know we are crossing a wrinkle-seeded plant, so that has to be little r, and then a pure round-seeded plant. So that is going to be big R, big R. If you see the word pure, homozygous, purebred, all of those mean homozygous or two of the same, as we can see from there. Now let's draw our Punnett square. So same as before, we set up our square. We put one parent on top, which in this case I'm going to use little r, which I'm going to put, again, anything that's on top comes down. Anything on the side goes over. And here is our resulting Punnett square. So let's find our genotype ratio. Let's first write down all the genotypes we have. All I'm seeing is big R, little r, heterozygous. So our genotype ratio is going to be 4 big R, little r. Now let's take our phenotype ratio. What does big R, little r tell us? Well, there's at least that one capital letter there. So that is also going to tell us that we only have the possibility of producing 4 round pea plants. There's no little r to be found. So we only have round as our offspring percentage. Sweet. Another example. Pause. I want you to try this one on your own, and then we'll go over it together here. Okay, check your work with mine. The letters themselves do not matter. I use T for this, capital T for 2 teeth, that being the dominant trait, and lowercase t for 1 tooth. The parents here, we have heterozygous, SpongeBob, and Sponge Susie is going to be purebred for 2 teeth. So the parents that you should have gotten, heterozygous for SpongeBob, and Susie is going to be purebred for 2 teeth, so she's going to be capital T, capital T. Let's draw our Punnett square here. There's that. Doesn't matter what parent you put on top. I'm just going to put SpongeBob on top and bring, again, anything that's on top is going to come down. Anything on the side, which in this case is going to be Sponge Susie, is going to come over. And there is our resulting Punnett square. So let's answer these questions. What is the probability that the children will have 2 teeth? Well, 2 teeth is the dominant trait, so as long as we have one capital letter present, then they'll have 2 teeth, which all 4 of our offspring do. So there's a 100% chance that the children will have 2 teeth. let's look at our genotype ratio. We have two different genotypes. We have big T, wrong one. We have big T, and big T, little t. And there are two of each of those. So our genotype ratio is going to be that. And finally, our phenotype ratio, we have to look and see what trait will an organism with big T have. They will have 2 teeth. What about big T, little t? 2 teeth as well. So our phenotype ratio is 4 offspring possibility, all with having 2 teeth. And our last problem together here is with Patrick. So, Patrick Starr has inherited a recessive disorder. His girlfriend is a carrier. So let's think about what those mean. First off, again, the letters themselves don't matter. I used N for this example, but recessive disorder is going to give us, we know that in order to have the recessive trait, you have to have 2 recessive alleles. Carrier, remember, means heterozygous, and so those are what the parents are going to be. Patrick is little n. His girlfriend is big N, little n. Now let's draw our Punnett square here and see if we can figure out what their offspring possibilities will be. So, going to put Patrick up at the top. Again, anything on the top comes down. Anything on the side goes over. Big N, little n, little n. And then from there, let's figure out our probabilities here, which will help us. So let's figure out our genotype ratio. The 2 possible genotypes we have are big N, little n, and little n, and there are 2 of each. So there's our genotype ratio. And our phenotype ratio, we have to look and see what these tell us. So what does big N, little n look like? That is going to also be a carrier, but still look normal. So we have 2 normal. And then little n is going to give us the actual recessive disease. So 2 normal to 2 disorder. Last thing here to fill out is look and see. What is the probability of having children with this disorder? Well, we have 2 out of 4. So that's going to be 50%, or 1 out of 2. And what is the probability of children as carriers? That is also going to be 50%. Because again, carriers means the same thing as heterozygous, just like that. Okay, so we're going to keep doing lots and lots of practice problems with these, so you will get very, very familiar. But if you need to, pause at any time, go rewind, look at these problems over. If you have questions, come talk to me. Thanks for watching.